Hi, my name is Elizabeth Sylvia,
And my name is Christopher Baker.
and today we're going to be talking to you about our research on the food choices in the solar-powered sea slug, Elysia crispata.
So, our project focuses on the chemotaxis of the saccharine sea slug, Elysia crispata. Chemotaxis is a type of behavioral response towards a given chemical stimuli. For the purpose of this experiment, cues transferred through the water column aid in food detection. Saccharines feed exclusively on algae, and many species exhibit kleptoplasty, which is the ability of an organism to sequester chloroplasts within their own cells to use for photosynthesis. Now, the objective of our experiment was to investigate how a period of growing starvation on Elysia crispata affects their affinity to algal-based cues from their known species of prey, Bryopsis plumosa. We hypothesized that due to the slug's photosynthetic nature, a delayed response to food stimuli will follow suit.
We collected our bryopsis every other week from Sand Key Park in Clearwater, and the Elysia crispata were collected from Key Largo last August in 2020. So we fed the bryopsis to the crispata for 2 weeks prior to the experimentation and the subsequent starvation. And we used something called a choice apparatus, which was three pieces of connected PVC. So the two peripheral chambers had either algae treated water fed in or control water. And we tested one slug at a time for an hour period and recorded their position at the end of that hour as either made algal choice, control choice, or no choice. So these trials occurred every 2 weeks over an 8-week span. And for data analysis, we used a chi-square goodness of fit test and Microsoft Excel.
And we would just like to point out that the data being presented today is preliminary as the experiment is still ongoing. We used 30 slugs and saw that most slugs did not make a choice during the experimental period at all. We also received, after statistical analysis, a significant p-value of 0.042.
So the results of this experiment are preliminary, and this is due to the fact that many of the slugs had become lethargic and or died prior to week 4 of experimentation, which did limit our data to week 0 and 2. So we did see an algal preference during week 1 as opposed to week 2, which was unexpected due to the photosynthetic nature of the slugs and the lack of starvation prior to this period. We also saw that more slugs made no choice on week 2, which may be due to the starvation induced lethargy kicking in, combined with a lack of motivation due to their photosynthetic capabilities. But all in all, we mostly saw that no choices were being made, which we did expect as, again, our data is limited to week 0 and 2. So as we progress beyond this time period, we expect to see some choices being made as their photosynthetic capabilities will begin to decline. So again, we're running this right now and hope to do it over an 8-week time period. And future work for this experiment may include measuring the rate of photosynthesis in the crispata throughout the same starvation timeframe. That's all from us. Thank you very much for listening.